move on to how twins are basically born. First of all, we have dizygotic twins, which arise from two eggs that are separately fertilized by two different sperms. So there are always two zygotes. And these will have two separate amniotic sacs and two separate placentas, basically two chorions. Monozygotic identical twins arise from one fertilized egg, one egg, and one sperm that splits in early pregnancy. The timing of cleavage determines chorionicity, which is basically the number of chorions, and amnionicity, which is basically number of amnions. So there's a cleavage which happens in 0 to 4 days, that's a separate chorion and amnion. Then there's a cleavage that happens 4 to 8 days, that's a shared chorion. Then there's a cleavage that happens at 8 to 12 days, and that's a shared amnion. And then there's a cleavage that happens after 13 days, and this is basically conjoined twins where the entire body is shared. Now, the mom's uterus basically, there is an amnion, which is the inner membrane or the amniotic sac, in which is the embryo and fetus and cord. And then there's an outer fetal membrane, which is basically the chorion, and that also includes your placenta. So the chorion is the placenta side or the outer fetal membrane, and the amnion is the water bag around the entire fetus. Now, dichorionic means there are two chorions and usually two placentas. Monochorionic means there's one chorion. Diamniotic means there are two sacs, and monoamniotic means there's one sac. So first of all, we have the dizygotic twins. In dizygotic fraternal twins, we have two eggs and two sperms, two separate zygotes, and two separate blastocysts. So it's di always. In monozygotic identical twins, the timing determines the sacs or the placenta. Now at day 0, when you're basically the fertilization is happening, you have a zygote, and day 1 to 3, the cleavage is basically creating a morula. Then day 4 to 5, the blastocyst forms with the trophoblast outside, and on day 6 to 7, the implantation begins. And then you have the chorion or placenta starting from the trophoblast. Amnion forms a bit later, around the bilaminar disc stage. Now, so if the split happens before a structure forms, each twin gets their own. And if the split happens after a structure forms, so the twins have to share it. Now, a split at 0 to 4 days is basically the time when the early cleavage or morula is forming. Right? So you get dichorionic, diamniotic, like fraternal, but genetically identical. So a dichorionic, diamniotic is basically just like the fraternal dichorionic diamniotic twins, these ones, the dizygotic fraternal twins are very similar to the dichorionic diamniotic, except that these are genetically identical because one zygote splits very early at 0 to 4 days, and two embryos are formed before the chorion forms. So they're basically di. Then we have a split at 4 to 8 days. This is the blastocyst stage. This is when you have monochorionic diamniotic. Why? Because the embryo reaches the blastocyst and trophoblast is present. So now a split that happens at 4 to 8 days, basically the chorion is already established. So it's a monochorionic. At 4 to 8 days, you know that the chorion is already established. So they have to share a placenta. But the amnion is not yet established. So these twins have separate amnion sacs. So this is when you get monochorionic, diamniotic. This is a 4 to 8 day stage, and this is when twin to twin transfusion syndrome is very common. It happens in monochorionic twins because they share a placenta with vascular connections. Then you have a split at 8 to 12 days after the amnion forms. Now, once the amnion is already formed, the chorion is formed, the amnion is formed. Now, a split will create monochorionic, monoamniotic. This is rare but risky. Why? Because now the embryo splits after the amniotic sac exists. So basically, the twins share a placenta and they share an amniotic sac. There's a very high cord entanglement risk because both cords are in the same sac. That is something you should know. After that, a split after 13 days is a very late split. You get conjoined twins. Basically, the twins share body parts, and this is sometimes described as incomplete separation. So basically, what kind of twins do you have? You have a dizygotic fraternal twins where you literally have two zygotes. This is a dichorionic. Then you have a monozygotic one where you have 0 to 4 days, but this is also dichorionic. Why? Because before the chorion is forming, you're doing the multiplication, the split, right? So basically, you have each twin has their own chorion and amnion. The only difference between this and the dizygotic one is that the monozygotic dichorionic diamniotic twins are genetically identical. How clinicians identify this on the ultrasound? In dichorionic, you have a thick dividing membrane, which is like a lambda twin peak sign. In monochorionic diamniotic, there's a thin membrane with a T sign. And in monoamniotic, you have no dividing membrane seen. So in di you have a safest twin arrangement. In mono di you have a twin transfusion syndrome risk because they're sharing a placenta. In mono you have a cord entanglement risk, and in conjoined it's a very complex anatomy because the organs are shared. We now move on to our next section, which is basically the placenta. What is the placenta? It's the primary site of nutrient and gas exchange. So let me go to the placenta page. Okay, so what's the placenta? It's the primary site of nutrient and gas exchange between mother and fetus. There's a fetal component and there's a maternal component. The fetal component has two layers. The inner layer of chorionic villi is basically the cytotrophoblast. The cytotrophoblast makes cells. Now, the outer layer of chorionic villi has syncytiotrophoblast. This synthesizes and secretes hormones like HCG. This is structurally similar to LH. It stimulates the corpus luteum to secrete progesterone during the first trimester. So, syncytiotrophoblast synthesizes hormones. It lacks MHC1 expression, so there's decreased chance of attack by maternal immune system. Now, the decidua bisalis. We will look at the decidua bisalis while we study these parts. Okay, so first of all, you have the placenta. Placenta is an organ where maternal blood and fetal blood come very close but do not mix to exchange gases, nutrients, and wastes. The fetal component basically comes from the fetus. This is called chorion frondosum, and this forms chorionic villi. This is the tree-like villi that project into maternal blood pools, which are the lacunae. The maternal blood pools are basically the lacunae. Now, there's a maternal component which comes from mom, and this is the decidua basalis. This is basically the endometrium of the uterus. The maternal side that contributes blood supply and forms the bed where villi attach. The decidua basalis is basically the bed. This is the decidua basalis. This is basically the bed where the villi attach. Okay, so basically these villi are entering the decidua basalis and they're attaching here. Now, here also you can see the decidua parietalis, the decidua capsularis, and this is the decidua basalis where all these villi are attaching onto. This is the chorionic cavity, the fused decidua parietalis, chorion, leaf, and amnion here, the placenta here. Okay, moving on the fetal side. Now, something to note here is basically now we have a placenta and we have the uterus. When we talk about the placenta, we have two layers that are important. The syncytiotrophoblast, which is basically the syncytiotrophoblast is the outer layer here, and we have the inner layer called the cytotrophoblast. The cytotrophoblast creates cells. The cytotrophoblast is basically the inner layer and it's mitotically active. It divides. You can think that this is the builder cells that can make more trophoblasts. But then you have the syncytiotrophoblast, which is outside. The syncytiotrophoblast is the outer layer, this one. The syncytiotrophoblast is the outer layer. It touches the maternal blood directly and it secretes hormones. It secretes the HCG. It invades the maternal endometrium here, is invading the maternal endometrium, and which part of the endometrium? We're invading the decidua basalis. So we're invading the decidua basalis, the villi is attaching here, and that is happening to establish the placenta. There's a major immune trick basically. This syncytiotrophoblast has little or no MHC1 or 2 expression. This is so the maternal immune system is less likely to attack the fetus. Now, the syncytiotrophoblast synthesizes hormones and it lacks the MHC expression, so there's no immune invasion. Now, we have maternal blood pools that are called the lacunae. Now, maternal blood pools, I'll show lacunae in another picture. Here, can you see this lacunar network? So the maternal blood pools lacunae, the syncytial trophoblast forms spaces called the lacunae which fill with maternal blood. Can you see this here, that this syncytial trophoblast here, it fills these spaces. Why? So maternal blood is literally bathing outside of villi here. It's filling these spaces, and these spaces are called lacunae. Maternal blood needs to bathe outside of villi, but fetal blood stays inside the fetal capillaries. Okay. Now, coming back to our picture where we could see the lacunar network, this is the lacunar network. This is also our lacunae, which are basically maternal blood pools. Now, exchange happens across a very thin barrier between the maternal blood in lacunae and the fetal blood inside the villus capillaries. They do not mix unless there's trauma, abruption, or delivery. But there's this little layer, can you see here? So basically, these are the maternal blood sinuses. This is the giant trophoblastic cell. This is the syncytial trophoblast layer. This is the basement membrane. And here we have the connective tissue, right? So what happens is that this is the embryonic blood vessel. This is the basement membrane. This is the connective tissue. This is the cytotrophoblast and syncytotrophoblast, and around these are the maternal blood surrounding chorionic villi, the lacunae. So exchange happens across a very thin barrier between the maternal blood in lacunae and the fetal blood inside the villus capillaries. They do not mix unless there is trauma, abruption, or delivery. Now, there's a placental barrier, right? What separates the mom and fetus? Basically, the syncytotrophoblast here, and then the cytotrophoblast here. You can see that also. The basement membrane, here you can see the basement membrane, and also fetal capillary endothelium, because these capillaries are inside the endothelium, right? These embryonic blood vessels are inside this endothelium. So as pregnancy advances, the barrier becomes thinner and exchange becomes more efficient. Now, what crosses the placenta? From mom to baby, you get the oxygen, water, electrolytes, nutrients such as glucose, amino acids, fatty acids, hormones, IgG antibodies, which provide passive immunity to the child, drugs, viruses. The torch viruses basically all get crossed, and from baby to mom, carbon dioxide diffuses, water goes away, waste products like urea, uric acid, and creatinine, and some fetal hormones like metabolites. So basically, here this is a little revision. You have the amniotic fluid, the amnion, and here is the chorion. This is the uterine wall. This is the placental disc, the chorionic plate, chorionic villi, the intravillus space, placental septum, maternal blood vessels, and this is where you're seeing all the circulation. So this is basically the fetal circulation, and this is the maternal circulation, and this is the basal lamina. This is the basal lamina, right? And this is the apical membrane. So here you have the syncytial trophoblast, cytotrophoblast, endothelial cells. This is the barrier through which the circulation gets exchanged, and this barrier becomes thinner and thinner as pregnancy advances. Now, placental hormones is a very testable question. The HCG comes from the syncytial trophoblast, and the structure is similar to the luteinizing hormone. The function is to maintain the corpus luteum early in pregnancy. The corpus luteum keeps producing progesterone in the first trimester until placenta can take over progesterone production. And if you remember, progesterone is also used as a contraceptive pill because your body thinks that you're pregnant, so it doesn't, the period doesn't happen, or the menstrual cycle doesn't happen. So HCG keeps the corpus luteum alive, progesterone stays high, and pregnancy is maintained. Other hormones placenta produces include progesterone. The progesterone maintains the endometrium and decreases uterine contraction. And the estrogen leads to uterine growth and breast development. And the human placental lactogen increases maternal insulin resistance to give fetus more glucose. Now, one more thing, the blood flow directions. This is important. The maternal circulation basically comes from the uterus to the placenta. Spiral arteries deliver oxygenated blood into intravillus space or lacunae, and the blood drains back via the endometrial veins. So that's your maternal circulation. These spiral arteries deliver oxygen into the intravillus space, and blood drains back via the endometrial veins. The fetal circulation is important. Now, from the fetus to placenta, how does this happen? So basically, you see umbilical arteries. There are two umbilical arteries that carry deoxygenated blood from the fetus to the placenta. And the umbilical vein is just one, and it carries oxygenated blood from the placenta to the fetus. The rule is that the arteries go away from the heart. So in fetus, umbilical arteries go away from the heart to the placenta, so they carry low oxygen. And the umbilical vein returns blood to the fetus, so it's high oxygen. So that's a little revision of. Another thing is when you get placental abruption or trauma, this can cause mixing of blood, and RH sensitization can happen if mom is RH negative. Otherwise, you can also get placental insufficiency, in which you get IUGR and fetal distress. And some infections and drugs can cross to the placenta and cause a congenital disease or teratogenicity. We move on to our next section, which is basically the umbilical cord, urachus, and vitelline duct. So first of all, when we talk about the umbilical cord, you know that these two umbilical arteries and the vein, they are sort of suspended in a Wharton's jelly, right? So the umbilical cord has a Wharton's jelly, and you have the allantois duct here, the amniotic epithelium, and two umbilical arteries and one umbilical vein. That's your umbilical cord. So, two umbilical arteries return deoxygenated blood from the fetal internal iliac arteries to the placenta, and one umbilical vein supplies oxygenated blood from placenta to fetus, drains into the inferior vena cava via the liver or via ducta venosus. This is a picture showing you the cord lining membrane here. This is basically the amniotic lining membrane. This is the allantoic duct, the umbilical artery, two umbilical arteries, one umbilical vein, and the Wharton jelly is basically the substance of the umbilical cord. So there's a single umbilical artery, it's a two vessel cord, it's associated with congenital chromosomal anomalies, and the umbilical arteries and vein are derived from the allantois. Now let's look at the little allantois that we're talking about. The allantois basically is the connection. It's coming from urachus, and this is a bladder umbilicus connection. So this is a connection between the bladder and the umbilicus. First, let's look at how the umbilical cord was traveling. So the placenta gives up the umbilical cord, which has the two arteries and one vein, and it goes. The umbilical vein goes through the liver, and through the ductus venosus, it gets drained into the inferior vena cava. Okay, that is something you should know. Now, the allantois is basically the urachus, and this is a bladder umbilicus connection. The allantois buds from the hindgut region and extends into the connecting stalk. It later becomes the urachus, which is a tube between the fetal bladder apex and the umbilicus. Normally, it obliterates and becomes the median umbilical ligament, a fibrous cord after birth. Now, here you can see the allantois, right?